Good morning, good afternoon, and good evening, depending on wherever you are. Welcome to our session number 40. This is a special series of COVID-19 sessions that we have dedicated to diagnostics and related issues in the COVID-19 era. My name is Anafi Mataka from ASLM, and I'm delighted to be your host today. Today, again, we have a special topic. We're going to be talking about genomic surveillance, which is a critical component to the epidemic response, exemplified by early detection, characterization, and tracking of these new variants, such as the 501Y discovered within and also, again, outside South Africa, and also other related variants, for example, the B1, 521, 525 discovered within Nigeria and other parts of the world as well. This is following also interesting sessions that came talking about the same subjects. We had Prof. Tulia here talking about the network for genomic surveillance within South Africa and efforts within the Africa CDC pathogen genomics initiative. Where we learned quite a lot. As we have done in the earlier series, where we brought in manufacturers of diagnostic products, we again today want to focus on technologies involved in these important initiatives. So today we are happy to be hosting colleagues from the industry, from Thermo Fisher Scientific, who are going to talk to us about what is it that they have on offer in terms of technologies around genomic surveillance. To do that, we would like to welcome Jashan Gokal and Gaina Thompson, who are application specialists from Thermo Fisher Scientific, to take us through the presentation. This will be a 1 hour long session that will be made up of the first part having a presentation, and we'll have a question and answer coming through. Please keep your comments and questions coming through into the chat box as the session goes, and we'll address them at the end. Remember, French interpretation, English to French language interpretation is available. Kindly go to your screen and choose the language of your choice. Colleagues, I help me welcome Jashan to take us through. Jashan, over to you.
So, as enough of you said, my name is Jashan Gokal. I'm a field application scientist with Thermo Fisher Scientific, and usually my role is to go into labs, train staff, install equipment, and make sure that everything is working as expected. Today, though, my role is to give you something slightly different, and it's to give you a brief overview of who we are as Thermo Fisher and how we support COVID testing and the solutions and offerings that are currently available to facilitate you in your research work. If I could just, yeah. So some of you may already know the name Thermo Fisher, and some of you might recognize some of our flagship brands or instruments and some of our reagents. Chances are, if you've ever worked in a lab, you probably have used something from us at some point in your career already. And for those of you who don't know us, we represent a world leader in science equipment and reagents, and these are equipment and reagents that we build around critical applications that are designed to support your research, no matter what area that research may be in. The coronavirus pandemic has brought us even further into the spotlight at the moment, and we've been responsible since 2019 for producing one of the first or the earliest COVID detection kits, and we're also responsible for equipping many labs across the globe with these test kits, with the equipment necessary to run these tests, and the knowledge and training for the staff necessary to respond to the epidemic. In fact, Thermo Fisher as a company has been responsible for producing over 20 million manufactured tests per week, and we have installed and, well, we're responsible for the most part for greater than 50% or over half of all the tests performed worldwide for the COVID diagnostic tests. And we have continuously developed and refined our products in order to suit you. So one of the things that you would probably be quite familiar with already, since we've been doing it for a while now, would be the actual COVID detection kit, the TACPath COVID-19 CE-IVD RT-PCR kit. I know that's a bit of a mouthful, but the workflow is pretty standard, and it's the one that I have on my screen at the moment. This is the workflow for the validated test kit, and it's one of the most sensitive and specific test kits on the market. It even allows for differentiation of mutations in certain cases, especially when you're talking about the UK variant. And this is specifically what we want to talk to you about today. The virus is mutating. It's mutating faster and in more ways than we initially thought. And the diagnostic workflow that we have, it needs help. And right now, what this kit is still valid for all of the variants that are out there at the moment, but we don't know what's coming next. And that's where the mutation and early detection panels come in. So remember that viruses mutate, especially RNA viruses like your SARS-CoV-2. And your SARS-CoV-2 specifically has been mutating at a rate of about 1 to 2 mutations per month in response to selective pressures of the environment that they're in. And it's really important to be able to track these mutations to determine exactly what we have, what we're dealing with, and eventually what it might become. The continued uncontrolled transmission of your SARS-CoV-2 is creating significant issues, not only for our communities, for our societies, but also for viral evolution itself. And through these mutations, these recently identified variants have acquired mutations so much more rapidly than we've previously expected, and in very, very uncertain ways. When it first started, or when we first picked up these variants, there were only 2 new coronavirus strains reported. It was the UK variant and the South African variant. And there were tons of news reports about these. In UK, you had over 5,000 cases of a single variant. In Portugal, 20 cases. In Italy, 17 cases. And all of these numbers have continued to increase. What's more worrying is that not only are the numbers of the cases increasing for these mutations, but the number of variants and mutants themselves have increased. Where we started off with just 2, just the UK and the South African strains, we now have about 5 and still counting. So because this is mutating so fast, and since these first 2 strains have been discovered, scientists have started investigating and really started looking for more instances of variation. And they found quite a few more. Who knows how many are still to come? And it's lots of key questions that still revolve around these new coronavirus strains that we need to answer. Among them is, are our detection methodologies still valid, and how can we detect new strains? And what are the origins of these new strains? How prevalent are they? How fast do they spread? And does this mean that the variants are of higher disease severity? Honestly, we still don't know. And a lot of ongoing research is needed to better understand how these viruses are evolving and spreading in the first place. It's so important for us to distinguish these new strains, to figure out their prevalence in society, and figure out, firstly, whether our tests pick them up, whether our vaccines are going to be effective against them, and exactly what phenotypic advantage these mutations and variants might carry in terms of epidemiology. Looking at global surveillance as a whole, this is one of the newer routes and one of the most prescribed routes for looking at epidemiological surveillance and epidemiological studies in terms of looking at where the virus has come from and where it could possibly be going. And it's important now more than ever because it's conducted to ensure that viral diseases match the reference strain and to track and monitor possible mutations. Since any change in the mutation, any change in the genome can impact public health policies, can impact opinions, it can impact how the virus spreads in populations. And a potential study of these treatment options and vaccine development research definitely hinges on whether we understand these mutations in the first place. There are many different ways of looking at this, and knowing that changes in the viral genome impact public health has put an increased pressure on us as researchers, as suppliers, as scientists who have some knowledge to find out a bit more to help the greater good. looking at these potential implications in some of these cases, we've seen an increase in speed of human-to-human transmission. We've seen changes in disease severity, evasion of certain vaccines, or certainly a reduced ability of certain vaccines to induce immunity. And we have seen in some cases of certain mutants being impacted by certain clinical tests. There are several coding mutations across this entire viral genome, and these several variants represent mostly within the spike protein, but we still don't know whether there might be potential for variants in other regions of this particular viral genome. And because a lot of questions have arisen around these issues specifically, we need to develop more sensitive methods for detecting the variants and these mutations in order to improve our tests from our side as diagnosticians. We honestly don't know what these impacts are going to be, but we can develop the tools necessary to enable our researchers to find out. So looking at global surveillance in a nutshell, what we're actually looking at are three interrelated but very important aspects of surveillance as a whole. What we're looking at firstly is strain surveillance, where we look at new and emerging variants that may arise possibly due to localized selection pressures of whatever environment these viruses find themselves in. And then we have your strain and mutation verification. And this is where we confirm that these mutations that we are discovering are in fact associated with disease and disease causation, and that we can look at the prevalence of these particular variants or these particular strains in society as a whole. And finally, there's a third aspect. And that third aspect comes in with your clinical diagnosis. And this is specifically looking at clinical diagnosis of your mutant strains in relation to its wild type, because this eventually will help us provide the correct treatment for that specific variant. Right now, the clinical diagnosis is most important and it's most essential because this is where we're going to get as much data as possible for us to understand how this virus is evolving, whether there are certain vectors at play, and how these mutations can affect the diagnostic kits that are used to test for them. And these three aspects should not be treated in silos. They're all part of one single workflow, and it's all done through very close ties between your research and clinical teams. Because although these are carried out by distinct technologies, they're carried out by teams with very specific and very specialized skill sets, and they are each carried out for a specific purpose. Each application is designed to support one another in the whole scheme of analyzing and understanding the virus and the pandemic that we're dealing with. So looking at these three aspects, and the two of them fall under mostly the research area, the research branch, whereas the one falls more under clinical, being the detection and diagnosis. There are three main technologies involved in this entire process. And this would be NGS, CE, and RT-PCR. And that's your real-time PCR, next-generation sequencing, and capillary electrophoresis, or Sanger sequencing, for those who are not too familiar with those terms. And really speaking, it's about using the right tool for the right job. You wouldn't use a hammer to break an egg. And although it can be done, there are probably a lot more efficient ways of doing it. Each one of these technologies and tools are used to their own maximum potential to achieve a particular result. And if we were to look at these three technologies in an absolute nutshell, what we're looking at are exactly what these different technologies can do and where they fall within strain surveillance, mutation verification, and RT-PCR detection or diagnosis. Your NGS specifically falls under the strain surveillance. It falls under looking for new species, new strains, and new variants. It's specifically important when you want to sequence a full or whole viral genome, and especially important when you are trying to detect new and emerging mutations. Your capillary electrophoresis or Sanger sequencing is a more targeted approach. It's when you need to target specific areas of that viral genome in order to interrogate it with an absolute high level of precision and specificity. And it's used for verification of these mutations, verification of the particular strains that you are working with, and in some cases, verification of your NGS results itself. And finally, you've got your RT-PCR. And this is for detection and mutation verification on the same instrument. So it confirms known mutations, and especially since this is a very targeted technology, it confirms what you know you're already looking for. It assesses emerging mutations and its impact on performance claims of new tests, and it can help us inform new product development. Looking at these three technologies, again, in terms of the general molecular biology workflow, you can see that they form three arms of the entire molecular biology spectrum. This is a very high-level overview. This is a very high-level workflow. But remember that when we start any kind of molecular biology application or any kind of experiment, our starting material is going to be nucleic acids. Whether this is DNA or RNA, it doesn't actually matter. But we need a solution or a robust system that can extract that nucleic acid for us. Once we have the nucleic acids available in a pure and highly concentrated form, what we're looking for is really trying to interrogate those particular nucleic acids, that particular DNA or RNA, for the genes, or in more precise terms, for a precise sequence within that DNA or RNA that is designed to tell us, well, something. And since the first step is nucleic acid extraction, we recommend the method that you use to be highly accurate, producing something that is pure and as high a concentration as possible, because this allows you to do anything. Once you have your DNA extracted, you can then take it into next generation sequencing. You can take it into capillary electrophoresis. You can take it into real-time reverse transcription PCR. And these are not just instruments. They represent entire workflows. Eventually, when COVID is over, and I am saying that quite optimistically, and I strongly believe that sooner than we expect, COVID will be over, these instruments can be used for anything else. They're not COVID restricted. They are not COVID related, locked. They can be used for any other kinds of applications. So the flexibility remains present. It's when you're looking at NGS, you can use the same DNA extracted from the same system in an NGS platform to look into pharmacogenomics or prenatal testing. With capillary electrophoresis, you're looking at HIV drug resistance or microbial fingerprinting. And with RT-PCR, well, this is something that a lot of people would be familiar with because it's what they're using to currently do your CIVD COVID testing. But that's just a presence-absence analysis application. There are lots of other applications like your viral load quantification, like your gene expression, that is still viable and still available to do on this platform. And what is nice is that your SNP genotyping as one application is exactly what the mutation panel is based on as a fundamental molecular technology. If we were to focus specifically on these research solutions for strain surveillance, the value of this is specifically to confirm the presence of these known or emerging mutations. And what we're looking at are these molecular fingerprints to assign specific mutants or genetic clusters to create a map to help us create an understanding of how this virus is spreading within your local communities across a nation and globally. And you can do this by either looking at the entire viral genome, or you can look at it targeted by looking at specific genes and specific regions within this particular genome. And this can be done either with NGS or capillary electrophoresis. And remember that the way that these work, they are different technologies. They're different tools. And they're designed for slightly different things. If you're looking for new variants, emerging variants, I would go for the NGS solution. If you know what you have and you just want to confirm what you have or just target specific regions, capillary electrophoresis might be the better way to go. So it's really a question of what do you need to analyze? When looking at whole genome sequencing as a whole, what you're looking for are the opportunities for the discovery and characterization of generally new viral, bacterial, and fungal organisms. But when you're looking at specific microbial or genetic structures, looking at the entire genome is an important step. And it's often the basis of any research project in molecular biology to go further. When looking at the SARS-CoV-2 genome in particular, there are entire kits and entire workflows that we can supply that already cover the entire genome as part of our ion ampli-seq research panels. And this assay is, for the most part, hands-off, automated, and enables you to go from that extracted DNA and RNA right into a full sequence of the entire genome in a single day. So there are lots of options, and it depends on specifically what you're looking for. These NGS systems are divided into different platforms. All NGS systems, at least, they can do the same things. What you're looking at in terms of its features are going to be how much of hands-off time you actually need in terms of its throughput, in terms of its automation. And something else to remember, or something that I have to reiterate again, is that these are not locked for COVID. Tomorrow you can turn around and use the exact same platform for any other kind of testing. Whether this would be fungal bioprocess, whole community analysis for IBS treatment, you can use the same platform tomorrow, and all it is a slight change of the kit that you're planning to use. When it comes to Sanger sequencing, the same rules apply. The technology's been around for a long time. It's an established workflow. And this technology can probably be found in a whole lot of your labs already. It's a cost-effective method for sequencing short targeted fragments and still remains the gold standard for obtaining any kind of DNA sequence. In the context of COVID, it's most likely going to be used for confirming your NGS applications or to sequence known mutations in your known genes. And there are lots of protocols and optimized protocols already available for us. With regards to your research solutions for strain and mutation verification, what you have is your capillary electrophoresis, but also your RT-qPCR applications, which are currently in development. Well, not really in development, they've currently been released, and these currently use qPCR, or real-time PCR systems that you are already have in the majority of your labs. And this is now a fundamental or a basic research technique that almost all labs have, and is the same technology used for diagnosis of these systems. When looking at TaqMan mutation panels, these are fully customizable, already optimized panels for targeting specific SNP mutations within your workflow, and this will give you results in about just over an hour. And the ability to distinguish between different SNPs to better understand their function comes through as pre-packaged, pre-optimized TaqMan assays and TaqMan kits, which are designed for direct usage with all of these platforms. This is a slight overview of everything that I've been talking about so far. I'm not going to go into detail on this slide, but it's a summary of what these technologies actually offer, where they fall under these different surveillance and verification platforms, and generally customers who are already using our TaqPath or similar kits will be able to adapt to this very, very fast. And again, as part of our mandate, we have a lot of knowledge resources available for you to use. We offer training content, we offer solutions and kits that are specifically designed to get you up and running as fast as possible. And these can all easily be found on our website with white papers, with blogs, with research updates, and that you can access through our website. And also remember that as a solution that we provide, it's about giving you the best value for what you need. And because of that, we have a whole lot of a team of specialists when it comes to these applications. We offer specialized training courses, and we make sure that you are up and running from day one, both for the instrument and for your training of your staff and engineering support that you need going forward. So from my side, thank you so much for your time today. Do you have any questions on what we presented so far?
Thank you very much, Jashan, for the wonderful presentation. So I don't know whether we can move on to questions and comments. Please keep them coming through into the chat box, and we will take them as they come through. Just to pick a few that have come through here, there's Amha Habden Michael, who is asking, do we have any data indicating the impact of the new strains from UK and South Africa regarding the current detection method, RT-PCR antigen detection?
So yes, we do. With regard to the detection method and the different strains, I can't speak for the new ones that are coming out, but for the existing, and I don't have any data for the Nigerian strain at the moment, but for the other strains, our kit still applies, it's still valid. And the reason for that is that the TACPath COVID kit was designed with redundancy. It's designed targeting 3 genes. So the one thing that we have seen is that it's fine for all other kits. It still works. It still shows you amplification of all 3 gene targets for all other variants except for the UK variant. For the UK variant, what you tend to see is a dropout of particularly the S gene, but the other 2 genes still amplify as expected. So what this effectively means is that using our kit, you can discriminate between the UK variant and any other variants that might be out there, because the overall specificity of this kit, because of the redundancy of 3 genes, still remains at 100% or close to 100%, but it now gives you an extra feature of discriminating the UK variant from anything else.
Thank you very much for that clarification, and also many thanks to Tseho Matatinyane, who also weighed in there, and I'll just read his response also in the comment. He says both the UK variant and the SA variant have the N501Y mutation in the receptor binding protein. However, the 2 strains are different. The N501Y mutation has not been found to affect the TACPath COVID-19 assays, nor have any other mutations in the 501 V2 variant. I'd like to call upon Dr. Skulile Moyo, one of the experts that is also presented on this platform. You can also chip in with your question around coverage. Dr. Moyo, over to you.
Yeah, I wanted to ask a question about your coverage for the S gene. Are you covering the entire gene? If not, what's your coverage percentage, and how many primers are you using? And also looking at the cost of your Sanger sequencing, how would you pay the NGS?
So with regards to the NGS costing, I will have to transfer that to my colleagues. But when it comes to coverage, it depends on the technology you're referring to. So with the PCR, of course, that's not going to cover the entire S gene. With that, you're looking at specific SNP mutations within your S gene itself. And there are also primers available and probes available for targeting SNP mutations in other genes. But that's not really answering your question. When it comes to the S gene itself, we do have 2 protocols available for covering the S gene. The one protocol uses 2 primer sets for Sanger sequencing, and this does a coverage of the mutation region for the S gene specifically. And there is another protocol available as well. So the one protocol gives you a full S gene overview, so it sequences the entire S gene. And the other one, and this gives you specifically discriminating between the UK and South African variants, because those changes are found in the S gene. And in terms of the primers there, you're looking at, I think at last count, there was 22 primers that covered the entire S gene with a lot of redundancy and overlaps, so that your base calls were as accurate as possible. I think each gene gave you, or there were about 6 different overlaps for each region in that particular, using those particular primer sets. So in terms of accuracy, quite high. And then if you're looking at your NGS specifically, that of course will do the entire genome at a very, very high, specificity. In terms of costing though, I think I'll have to defer to my colleagues for that.
All right, so normally we would try to leave the costing part in these platforms and maybe just concentrate on the technical bits. So I would like to ask if we can move forward on that part. I would like to take another question from Amha again, who is talking about the preference. What is the preference for capillary electrophoresis for targeted sequencing? Can we do the same with NGS? So that's his question.
So yes, it entirely depends on what you're looking at. Remember, the right tool for the right job. If you're looking at the entire genome, I would say NGS is your better option. But when it comes to targeted sequencing, when you already know what you're looking for or looking at, CE or capillary electrophoresis might be the better option. And that's just because of when it is targeted, and this of course depends on how many targets you want to look at simultaneously or how big the gene region is that you're trying to look at. Your capillary electrophoresis tends to work out slightly more cost effective, and you can fit generally slightly more samples on a particular plate. So if you're looking at, on most of our platforms, for a single read in a single direction, you're looking at about 700 to 800 really good base pairs, or a contiguous read length of about 700 to 800 base pairs. And by having multiple primers, having multiple overlaps, you can extend this quite a bit.
Thank you. Thank you very much for the response. And many thanks again to Vim. Thank you for joining. Vim says, FDA posts information on impact of SARS-CoV-2 mutations on test effectiveness. And he has also shared the link in the chat box. So many thanks again for sharing. This is one beauty of this platform. And also many thanks to Janine Kotze for also sharing links on how you can find more information on NGO solution for SARS-CoV-2 research. Let me give an opportunity to Matthew Isona to also chip in. Matthew, are you able to unmute and pose your question? Matthew, go ahead. Yes, you should be able to speak now. We can't hear you, Matthew. So I guess whilst he's trying to fix his connection, you mentioned earlier on, Jashan, that this is mostly, if I got you right, when you started your presentation, you mentioned that this applies to research, to your research settings. Is it always going to be a research, given that there's a call to make sure that we sort of increase, I think, our surveillance mechanisms. How do we start? I mean, even if it's starting simple and establishing routine surveillance, but the moment you talk about research, we sort of become a little bit scared to say that may actually call out for a lot. What
Yeah,
would you
I
add?
fully understand. And I do understand your concern with that. It's very easy to always talk about research, but the honest opinion is that one hand feeds the other. Without the research and the knowledge coming out of research, developing the diagnostic kits specifically become very difficult. And it's also very much a point of how many samples do you need to run through and what exactly are you looking at? When it comes to diagnosis or the diagnostic setting, specifically when you're trying to produce a result for a patient, what's more important than sequencing the whole genome of something that you have is just figuring out which variant you need. And when all you're doing is figuring out which variant you need, perhaps CE sequencing or a real-time PCR approach would be a lot more cost-effective, would be a lot faster, and would give you an equivalent amount of data or enough data to give you an answer to a clinician or a patient. When you're looking at the research arm, what we're looking at a bit more is trying to actually understand a little bit more about this virus and these mutations and where it's coming from. So the research arm very much supports the clinical and the diagnostic arm, but one feeds data into the other. And it's very important to have a good balance of both existing within your territories. Just so that, you know, if you do see an outbreak from the clinical side of a particular variant, you're able to take this knowledge or this data back to the NGS or to the research side and possibly determine where this outbreak is coming from. Taking it one step further, and maybe I'm looking 2 steps further in this case, where there have been instances of particular host genome responses to this particular infection. So if we understand what the host genome actually is, we're able to treat it a bit better from the clinical side, and understanding that becomes pharmacogenomics from the research side. So it's a very difficult question to answer conclusively, rather than saying it's good to have a balance between both, and one definitely does feed the other.
Thank you very much. Once again, Matthew, I've seen you. We'll come back to you just shortly. Let me take this question from Dignity Madziva, who says, with such high levels of gene mutations of SARS-CoV-2 variants, the non-targeted methods will be the best way to monitor mutation trends in the population. And then surveillance as we do the diagnostic. I think it's more of a comment. What do you say about it?
No, I definitely agree. When you don't know what you're looking at, the easiest thing is to look for everything. So yeah, definitely agree with that.
Thanks. I want to go back to you. You tried to come back, and I've also acknowledged Sajid. So Matthew, are you back? Let me.
Yes, sir. Thank you so much. Can you hear me?
Yes, go ahead, Sajid.
Yeah. Sir, my question is that the failure of S gene and the other 2 genes positive in a PCR indicates the UK variant. How can we detect the other 2 variant, the South African as well as the Nigeria or Brazil variant on PCR based assay? Is there any kit or method available?
So yes, the last panel I was referring to would be the SNP genotyping or mutation panel, and this would particularly target individual base changes or individual mutations within your specific targets. It doesn't only target the S gene; it targets all of the known mutations across the different variants that we've come across. I I'm sure that it does everything up to the Brazilian variant, which is the P1. And I think the Nigerian variant is still a bit new, so we haven't got anything on that just yet. But I'm sure this will be developed soon. The TACMAN mutation panel, if you have a qPCR system or real-time PCR system, works in exactly the same way as standard real-time PCR with your primers and probes, except that now your probes are specifically targeting your SNPs of interest, and you'll be able to discriminate between the different variants based on the probes that you have.
Thank you. Thank you very much for responding to that. Matthew, please go ahead. Thanks.
Yes, thank you so much for this presentation. I stumbled on it by mistake. I'm really happy to be part of it. And again, I just want to thank Jeshan for an excellent presentation. But I have just a little concern when it comes to the mutations that are based on the spike protein area. As we all know, the spike protein area of this virus is the variable region of the virus. So that implies that there is a high rate of mutations going on already. So I don't know if I missed it, but designing a real-time RT-PCR assay based on the spike protein might not be long-term, might not be the long-term goal to look for variants. Reasons, because today you might have one mutation and the next day you have another mutation, and the assays will keep on failing as new mutations are coming up. So in my opinion, the best approach should be target-specific sequencing, which is Sanger sequencing, and a real-time assay, which is based on the nucleic capsid protein, which is conserved among these viruses. But the question I really wanted to ask is, how often do you guys update the real-time assays that are based on the spike proteins or on the virus mutation? Because in an African context where the resources are not that much, we want to maximize the way long-term, we're looking at long-term capabilities, not really short-term capabilities. I don't know if that makes any sense to this panel.
No, thank you for that question. That was a great question, actually, and I fully agree with you. It's, especially within the African context, we just don't know enough about what mutations are happening and how fast. So you're right, sequencing would be the best option, especially for a long-term solution. For a short-term solution, yes, looking at individual mutations might work. And there are a lot of individual mutations that we can look at. So I think we still have time before all of those mutations are completely wiped out and replaced with new ones. But one of the nice things about the kits that we have is that not all of those mutations are specifically like A to C's or G's to T's or whatever. It's a single base change, but we do design these primers and probes in with some form of degeneracy as well. So it will pick up a base change for those particular regions for some of those assays. Having said that, I still agree with you, and CE is still probably, or even NGS would be a better long-term solution, especially with the amount of techniques or applications that you can do with these instruments long-term, even after COVID. In terms of your question about how fast these are updated, I don't have an exact time frame for how fast the assays are updated, but when we found out about the UK and South African variant, which were the first ones out, the R&D team responded within a period of about a month that they released an early beta test phase of the TACMAN mutation panel. And 2 weeks after that, we heard news of the P1 variant, and they released a TACMAN mutation panel for that particular P1 variant as well. So it just depends on how much of data we can get. And now that I have everybody on this platform, this is an appeal from us to you guys. If you see anything interesting, let us know, because only by getting data can we develop something new.
Yeah, thank you, if I may. Thank you so much for that clarification. I don't know if you guys have looked at—
one more question, Isona, so that we can move to the next ones. Yeah.
Yeah, I just want to ask just one more question. I don't know if you guys have looked at the mutational rate for this virus, because that can give you, for these specific S genes, because that can give you an idea of when you can update the primers moving forward. Thank you.
Thank you, Matthew. Yep, so there is, I believe, one in development, but it is still in development at the moment. So I'm not sure when it's going to be released, but yep, you completely hit the nail on the head with that.
Thank you once again, Matthew, for the excellent questions. Keep the questions coming into the chat box. We find these quite valuable so that we bring the industry and the users together. Again, these questions should be purely technical. We do not want this session to be more of a marketing platform instead, but purely a platform for exchange of information. So as I have one quick question, which is more of a follow-up to our previous special COVID-19 session, where we looked at the role of LIMS, the laboratory information management systems. And I'm thinking, having presented the various technologies that are available via Thermo Fisher, how do the existing maybe platforms for data management, how do we support data analysis, sharing, and maybe interpretation based on what you have presented?
So that's a good question. Most of these platforms come in with their own software. Generally, a lot of the software that comes through from us is also integrated with the cloud. So we do have desktop versions of software, cloud versions of the same software. So you can analyze your data on the go, through the internet, through your cell phones if you want as well. And one of the features is that it does have an export capacity or an export capability that exports as a CSV. So you can directly integrate it with your LIMS system, or maybe indirectly integrate it with your LIMS system. Would be a better way of saying it.
Thank you very much once again. I have more questions coming into the chat let me take this one from Dawit Ademi. He or she says, regarding application NGS for microbial sequencing, what is the advantage in getting results, in getting faster results and better outcomes?
I think it depends on the application. So I think when speed becomes most important is when it is for a diagnosis. So yeah, by having a microbial profiling, a simple example would be something like picking up a blood infection when going to a hospital. It's very important to figure out what organism is causing that infection and then choosing the correct antibiotic to treat it. So in terms of speed and why you would need that, I would definitely say diagnosis for one. And generally hands-on time or hands-off time becomes very important with that as well. You want to free up your lab staff to do as much as possible rather than having them tied down to one particular sample for an entire day. So it's a combination of what you're actually looking for versus how much of data you want to get out, I suppose. I'm not sure if that fully answers your question though.
Thank you for attempting that as well. Let me also take the opportunity to also acknowledge some posts and comments of appreciation from Sereke Tefashion, who says, thank you for the wonderful presentation. Thanks a lot, Sereke from Eritrea. And the same is also shared by Mohammed, who is also acknowledging the interesting and scientific debate that that should continue on this platform. One additional question that I would also like to bring upon here is sample collection and shipment. What can you say and recommend, particularly when it comes to the issues of surveillance as well?
I think sample collection is probably the most important question you could have asked, and it is probably the most important aspect of any kind of molecular biology workflow. You can have the fanciest equipment and kits downstream, but if the sample that you've collected in the first place isn't too good, the results may not be as good as you expect. So sample collection is definitely, in my opinion, probably the most important aspect of things. And the rules for sample collection are, universal generally. It generally doesn't matter what sample you're collecting, the rules are always the same. You keep the sample as pure as possible. You maintain a cold chain, or you store it in a solution that's designed to preserve whatever it is that you're looking for. So in terms of COVID's an RNA virus. When you're collecting it, most of the time you use the nasopharyngeal or nasal swabs. We actually have some kits available for taking a saliva sample itself. So no more of those horrible nasopharyngeal swabs which hurt like hell. I've had a few of them done and they're not fun. But saliva samples are perfect, but the kit is designed specifically to preserve whatever viral samples are coming through in that saliva. It's designed to stabilize it until your extraction can be done. And remember, so your first sample, your initial sample is the sample from your patient. It's the swab, it's the sputum sample, it's whatever, until you get to extraction. Once you get into the extraction phase, your sample that goes into the rest or your downstream molecular biology workflow is actually DNA or RNA. So making sure you have an extraction kit that purifies your DNA or RNA effectively and then preserves your DNA and RNA is also important. So definitely choosing a good kit for extraction, a good swab is very, very important. So yeah, definitely agree. Sample collection is paramount.
Yeah. Important indeed, and as the usual saying goes, garbage in.
Definitely.
So they say. And Kate has a follow-up question on that. Kate Slimani from the CDC Atlanta. Thank you, Kate, for joining. She said, for saliva, do you recommend neat saliva without preservatives or additives?
So the kit itself comes through with, it is a self-contained kit, and the input is actually just neat saliva.
Great. Thank you for clarifying that as well. I know we are almost reaching the end. You touched an interesting subject, which was also a key main theme in our last LabCorp session, where we talked about possibility of diagnostic integration. What happens after COVID? Can we be able to test other, other diseases beyond COVID? I just wanted to ask you again to clarify that. What exactly did you mean when you talked about the ability to test other organisms beyond the SARS-CoV-2?
So I know everybody is so focused on COVID right now that we tend to forget that the equipment that we have, it's just that. It's equipment. It's a tool that's designed to look at, in this case, DNA. And really speaking, it doesn't matter where we get the DNA from. Your equipment's still going to look at it. So here you can look at in terms of this equipment, in terms of next generation sequencing, your input is DNA. And whether that's human DNA analyzing your metabolome or transcriptome, whether it's microbial DNA coming from a complex environment such as a mine, where you could look at individual organisms responsible for acid mine drainage. All of these instruments would do the exact same thing. In terms of your capillary electrophoresis, one of the most common applications for it is HIV drug resistance. It's where it does, in terms of the way that it works, exactly the same thing as looking for a COVID mutation. It looks for an HIV mutation, and it just aligns that particular mutation against a known database, and then gives you an output where it tells you whether this patient is resistant or susceptible to a particular antiretroviral treatment. As long as you can get DNA in, the world is your oyster. You can imagine the possibilities when it comes to these platforms. And the majority of them are open platforms as well. So as long as you can find a kit for it, you can probably achieve it.
Thank you. Thank you very much for summarizing that as well. And also to acknowledge, I think many of our colleagues from the industry, COVID-19 showed us that we can work together to conquer. We still have a fight to go, but I'm very confident from the diagnostic front. I think we are trying. We are trying. Hopefully, we continue to do that, and hopefully, our best would be enough. I can point out examples in certain countries that I will not mention right now, where the initially so-called, in quotes, closed platforms could actually were actually opened so that they can actually test for the SARS-CoV-2. So many thanks actually for that gesture as well, and moving forward. Jashan and maybe Thompson, I know she joined later on. In a minute or so, any closing remarks?
So for me, I would just like to thank everyone for joining us today. I know this was a brief presentation, and definitely thank you for all the questions that came through. It's good to have a scientific discussion like this. We need to do it more often, especially because there are so many different players involved in what's going on at the moment that we all really need to be on the same page in order to achieve the same goal. So thank you, everyone, and continue doing the good work that you're doing. And if you need any help technically or otherwise, please let me know.
Thank you very much, Jashan, for the wonderful presentation, and thank you all for the very engaging discussions. I can see quite a lot of messages acknowledging the scientific debate and the scientific information that we managed to share today. And we also glad that you continue to find these very useful. On your screens right now is a code for you to be able to download your certificates should you need any certificate to show that you were with us for the past 1 hour. We will continue to bring these sessions to you. Tomorrow, we also have another interesting session that is going to introduce and discuss one of the innovations that is coming through in trying to get the world back to normal. The issue of the trusted laboratories and the travel passport. So watch out for, I think the invitations have already come through in your inbox. So same time tomorrow, please tune in, and we have also another great session. Till we meet, it's goodbye for now. Thank you.